morning. Assalamualaikum warahmatullahi wabarakatuh. Welcome to the series of Unique First Hybrid Webinar, which today will pick up the topics about tropical medicine and neuroscience. Thank you very much for all the attendance. Okay, first of all, I'd like to welcome to all the speakers, Professor Irawan and Professor Roger Frutos, to our special guests and the representative of Biomedical program, Faculty of Medicine, Public Health and Nursing, Universitas Gadjah Mada. Okay, before we start to our main session, let's hear the opening speech, which will be delivered by Dr. Junaidi Yunus, the Secretary of Master Biomedical Programs. Okay, please step forward.
Okay, thank you so much. Okay, good morning, ladies and gentlemen, the Honorable Prof. Rika, and also our distinguished speaker, Prof. Irawan, and also Prof. Roger, and also our distinguished moderator and all participants. Yeah, thank you to each and every one of you for being here today, at this wonderful place. My name is Junaidi Yunus from Department of Anatomy. It is such an honor for me to speak on behalf of the Dean of the Faculty of Medicine, Public Health and Nursing, and also the organizer. Let me begin by giving you a warm welcome to this webinar, which is organized by Master Program in Biomedical Sciences. First of all, let us express our gratitude toward the Almighty, whose blessings are always with us, so that we all can be here in good condition and health. And we would like to express our appreciation also to our distinguished speaker, Prof. Irawan Satriotomo from University of Florida, and also Prof. Roger Frutos from Montpellier University, who could join us to share their valuable expertise. And this webinar has the title Biomedical Unique Versus Virtual Education Research Exploration Series, where the current knowledge and also development related to the topics will be presented in this session. Prof. Irawan will share current knowledge of the role of neural and vascular crosstalk in neurodegenerative diseases and nerve repair. And later, Prof. Roger will share current knowledge of the pathogenesis, pathophysiology, and prevention of dengue and Aedes-borne disease. Once again, welcome and thank you very much to all the distinguished speaker, moderator, and also all participants for attending this event. We hope your participation will be a valuable learning experience. And the last, by saying Bismillah, we officially open this webinar session. Thank you so much.
Okay, thank you very much, Dr. Junaidi Yunus, for the opening speech. Okay, now we're moving on to the main session of our discussion that will be organized by our moderator, Dr. Dian Eureka. For the Dr. Dian Eureka, please, the stage is yours.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen, and also Honorable Professor Rika, Professor Roger Frutos, and also Professor Irawan Satriotomo. Okay, so for this hybrid webinar, we will have two sessions. The first session, we will have a talk from Professor Irawan Satriotomo, MD, PhD, from University of Florida. He will give a talk about neural and also vascular crosstalk, role in neurodegenerative disease and repair. And then after that, Professor Roger Frutos will give a talk about the dengue and Aedes-borne disease, how they work and what do we do. So for the first session, maybe before Professor Irawan Satriotomo give his talk, I would like to introduce him. So let me read his CV. Okay. So for Professor Irawan Satriotomo, he is a lecturer in University of Florida. He is also the founder of Tissue Protect and also Tissue Protect Exomet Therapeutic in USA. So he graduated. He's a medical doctor from the Universitas Gadjah Mada, and then he graduated his doctoral degree in Kagawa University in Japan. Okay, and then after that he moved to USA. Okay, so for before Professor Irawan Satriotomo, we would like to invite you to give the talk. So, Professor, the stage is yours.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. And first, I would like to thanks for the invitation, and also I'm glad I met my teachers and also my colleague now, Prof. Rika, and also my colleague, Dr. Rogers. Today talks, actually, the organizing committee asking me to do, first slide, please. Yeah, giving the talk about the neurovascular crosstalk and role in neurodisease and repairs. I do some study using the stroke models, so probably I will touch in base about the base of the neurovascular crosstalk, and then I will showing you some data that we have and we did in our lab. So if you're talking about the neurovascular unit or neurovascular crosstalk, we do have some what we call the neurovascular unit. Yeah. It's a multiple cellular system that consisting neuron and also parachute that is very different with the others blood vessel. And also we do have the smooth muscle, glial cell, endothelial cell. So what the function that they do in these our brain, they have what we call the blood-brain barriers, and they only can crossing the small molecule. You guys need to remember this. And only a selected nutrient can be transported into that system. And also they have function as a maintenance vascular impermeability, as well as the neuroimmune surveillance and brain hemostasis. So as I mentioned before, that only just a very few small molecule can crossing, mostly it's below 500 Dalton. And I just talked with Prof. Rika that now the technology can break down the cell into a small unit, what we call extravasculars. And one of the unit we call this is exosome. It's, the size is between 60 nanometer until 300 nanometer, and it still contain what the cell itself has. So now people try to harvest that like a cell or stem cell to get that what they have in the stem cell, like growth factors, messenger RNA, and everything. So the other function is a neurovascular coupling that is maintaining the cerebral blood flow, regulation of energy supply, as well as the hemodynamics. Yeah. Next slide, please. So yes, if you see how the neurovascular develops, so during the embryonic 8.5 to 10 days, So what's happening is that there's ectoderms, and also they have the mesodermic that contain of the what we call the deploying very vascular plexus, and then they will try to infiltrate that tubules and then make the vascularization in that area or in that region. And it's quite different between the spinal cord and also the forebrain. Yeah, you guys can see the differences how they try to make a vascularization inside the tubules. And then if is happening, and so we do have the what we call the combination between the vascular system in the plexus. So we have radial glia, basal progenitor cell, or migrating interneuron that try to putting the cells inside inside the nerve system. Next slide, please. So again, oh, sorry. Okay. So again, we can see this the parasite, as I mentioned to you, is quite different with the others blood vessel that is maintaining the integrity of the cell itself. Yeah. So they have system that also communicate with other cell like astrocytes, and also they have when during the neuroinflammation process, for example, they can divide it into the migrating cell like macrophages. Yeah. So this is the diagram or the cartoon that you can see during the process. They can sense the inflammation process and modulate the microglia properties. So they can become the anti-inflammatory microglia as well as the pro-inflammatory microglia. It depends on the situation and also the phase of the disease process. For example, during the acute phases, mostly they go to anti-inflammatory microglia. But then during the long process, they will changing the system become pro-inflammatory. Yeah. So how they do that? Yeah. So mostly, as we know, during the process, for example, like a stroke or other inflammatory process, so this can make the condition become very ischemic. So then what's happening with the stem cell property of the parasite, they can differentiate into microglia. Yeah. So this is just the what we call the vascular signaling. Then we can see that they try to regulate the regulation of the flow in the brain as well as the BBB, blood-brain barrier, as well as the immune surveillance. And there is a lot of actually signaling that include during this phase. Yeah. Between the neuronal and vascular, there's communication, and mostly you guys can see that's conducted by the vascular endothelial growth factors. And the interesting thing is with the vascular endothelial growth factor, they have vascular endothelial growth factor from A to E, and each vascular endothelial growth factor have their different function. And before we thinking, because it's just for the migrating or the factor for developing a new vessel, sorry. But now we know that this is not only for the purpose of the extended or the new vascularization, but also actually they have other function as a trophic factor to growth factor for others, like a brain, I mean the neuronal and others purposes as well. So during the lifespan, so as we know, there's a lot of things going on, like a gene of neurodevelopmental disorder, and also during lifespan, they have many, like a brain trauma, mood disorder, or psychiatric disorder, and everything. Then this will changing the integrity of this vascular system, and if it's getting faced to the aging process, that we know there's like a cognitive decline or dementia or neurodegeneration, this make a profound the function of this crosstalk. So we know that endothelial dysfunction is altered by the time. Then it can impair, like alter the cerebral blood flow or hypoperfusion and everything. So then what's happening is aging process, traumatic environmental stressors, or other genetic neuroinflammation can induce the neurodegenerative diseases like autism, schizophrenia, multiple sclerosis, and other diseases as well. So this is what I mentioned before. Like the macrophages or the astrocyte is actually the glial cell that people thinking that have like a bad effect, but during the healthy state, they have a lot of function, like maintaining the antioxidant, and also induce the trophic factors, and also ion hemostasis like this. And the morphology also quite different during the healthy state or the pathologic stage. You can see the cell size is quite small, and also they have more ramification. But then when there is a inflammatory process or a traumatic during the process, so that they will change, become the more reactive, and you guys can see that the size of the cells become more larger, and the processes of the cell also become more bigger and more longer. And this is actually is quite interesting, because one of the paper that I published in a brain research journal showing the model of the enucleation. You guys know the enucleation? It's really in the nutrition. So what I do is I took the eye, one eye, and just try to see how the other part of the eye is compensate the mechanistic. So different with the human, when we can see the eye, so we only have some spaces that, but the rat or the rodent actually have more like 180 easily, so they can see you from the back as well. So meaning what? Meaning the expansions of the neural system is also different. But the interesting, when I cut one of the eye, all the part of the collicular superior cell, the neural, actually very sparse. But then the glial cell, the reactive astrocyte, in this case, is have prolonged processes that make communication between cell to cell. So this is what they said, that actually the communication of the cell during the degenerative process is helping by the glial cell itself. Just this is what we're thinking about how the Alzheimer's or dementia occurs during the processes, and how it's involved in the brain injury or the vascular injury. So beta amyloid or neurodegenerative process is actually is a part of the vascular insufficiency because of the time or the aging process and everything. Then it's make the neural become dysfunction and death. Yeah. So it's also can induce a lot of things like the white matter damage or others, hypermetabolism and everything that can induce also vascular cognitive impairment, then lead to the Alzheimer's diseases. I'm sorry for this very small table. So, but I would like to showing you guys that there's a like autism, yeah, autism spectrum disorder or schizophrenia, multiple sclerosis like Huntington disease, Parkinson or Alzheimer's disease is really associated with the neurovascular unit. So it's led by the altered by cerebral blood flow, as well as altered with the BBB itself, the blood-brain barrier itself. So what's happening during the, for example, diabetes mellitus? So during the diabetes mellitus, we know that there is the hyperglycemia as well as the hypermalsemia, or also there's some oxidative stress as well as in the inflammatory process that can change the neurovascular unit on that blood-brain barrier or the endothelial system as well. And this makes the microglia become more abnormal, yeah, as well as the oligodendrocyte also become dysplasia, so they cannot extend it more. Then this is make also pericyte become abnormal, and then induce the neuron become more insufficient to get the energy uptake. You guys can imagine, because the vascular is needed for the energy as well as the oxygenation. So this is induce the neurodegenerative diseases. Yeah. And also you guys can see that not only just neural, but also others glial cell also involved in that processes as well. Again, this is like as I mentioned to you, there is a normal cerebral blood flow is appropriate for maintaining the cognitive dimension. But if during the shortfall coupling because of the disease process, they induce the neurovascular dysfunction and also motor dysfunction itself, then it can lead a lot of things like a cortical macroinfarct as well as amyloid beta and tau neuronal inclusion. This is one model that we do using the stroke models. So it can be ischemic or hemorrhagic. And mostly the ischemic take a larger part of the ischemic stroke, like 85. And then also the hemorrhagic have a small numbers. But the problem is the hemorrhagic stroke mostly the mortality is much more higher. And we can actually mimicking this model using the animals. Sure. So the ischemic stroke again is a third leading cause of the death, but also in Asia is also number one. The hospital stroke hospital in Cawang, Rumah Sakit Pon, now has not only patient with the age of 50s, but now because of the lifestyle and everything, we found that there's a lot of new cases starting from young ages. This is very probably because of the lifestyle. Then it's a count of 1 to every 18 death in USA. And the problem is the medical cost for this stroke is really very large, including in Indonesia as well. So, and the risk of the stroke is increased by the aging as well. So we know that we can mimicking this model, so we know how we treated that model. And until now, there is no drug that can be used for treated the stroke. But we only have tissue plasmin organs for the ischemic stroke to protecting the ischemia become more larger. So actually the function or the drug, like a TPA or others, is to make the core, the red one, is stay in the size, and not the penumbra side, the white one, not expanded because of that processes. So it can prevent from the minutes until the delay of the weeks. And the problem in here is the time. The TPA only consider up to 4 hours. Below that point, there's no effective drug to control the ischemia. So and you guys can see per stroke, it is meaning by the time it is happening, we lost like 1.2 billion of neuron. Per minute, we count 1.9 million, and per hour it's already 100 million. So this is a really very speedy process of the stroke itself. So what's happening during the stroke? So we have no ideas how the pathology is happening, because there is a lot of theory behind this pathology. People thinking of the progressive neural injury, because the neuronal cell death, ischemia induced in cortical, subcortical, and also delay make a cell death and white matter injury. And then again, will induce the astrogliosis or reactive astrocytes and microglia, and make a glial scare formation. And there is a lot of mechanistic that we're thinking. Most of the famous ideas, glutamate excitotoxicity and free radical production like RAS. But we still doesn't know. When we try to inject antioxidant or whatever, it's still not have good result in a couple study that people did, and we did as well. So how the complexity is in the stroke injury is progress. So you can see during the 6-hour time window, we found that there is already energy failures, because the blood flow is stopped and everything is happening, and it will induce the excitotoxicity. And the other thing is I'm really sorry, I would like to showing how the BBB actually opening. BBB opening starting from the 12 hours. So what's it mean? Everything now is dispersed, and then also they cannot get intact. That make the core become expanded to other area and induce the cell death in many area. And then it will be lead the activation of the microglia as well. And then the other problem also, the blood-brain barrier also will degrade it, and cytokine or chemokine will induce more death cell in that region as well. So again, this is not only just in the brain what's happening, but also the other part of body as well. So it can lead to multiple organ failure as well. So it can lead the cardiomyopathy, can also changing the gut dysbiosis, as well as induce the pneumonia. Yeah. So but the other interesting point is now probably you guys hear about the neuroplasticity. So during the stroke or during these processes, actually there's also plasticity also happening as well. Yes. One of the plasticity that happening is in the spine itself. So you guys can see the dendritic. The dendritic actually has the very small what we call the dendritic button. That is the where the synapse is connecting to each other. During the pre-stroke, you can see that there is a very tiny and a lot of branches of the dendritic with the button. But when ischemic happening, so they will disperse, and then you see there is no connection between each other. But 6 minutes later, that they come back to connecting each other, and 90 minutes later it already repairs. But again, this is model we use in rodents where we know that the small mammalian actually very plastic, but in human we still try to investigate it more further. But if you see also the connectum, this is what we call connectum. So we use the functional MRI and see that how the fibers are crossing. So you see that the part of the brain that lost will try captured by the other area that's still intake. So this is the paper from Murphy did in 2009. So if you see that there's one part of the ischemia happening, so the other part of the relay will take over. Yes. So again, there's a lot of candidate for therapy from the neuro, microglia perspective, endothelial or astrocyte. And also there's many theory about how the concept of ischemic injury and acute stroke management happening. But until now, again, we don't have any good result or good drugs to prevent the stroke to not get expanded. So what we do is we try to see the meta-analysis study, and then we found that there's a lot of study using hormonal therapy like estrogens, until also the other anti-inflammatory drug and also antioxidant. One of study that we did actually is we use the estrogen. The idea is we know that there is very few incidents of the stroke in women premenopause, meaning before menopause they don't have a stroke. But when they get menopause, so now the incidents were higher. So what we're thinking is because the women with before premenopause have the progesterone and estrogen higher than the postmenopause. So that's why we don't find it. Yeah. And that's why when we try to using the estrogen in this model, and again, one interesting thing, we cannot use the stroke model with female rodents. If you guys should try to use that, the infarct size is very small, or they don't have any. Why? Because the estrous cycle of the rodent is very speed. It's like probably around, if I'm not mistaken, it's 3 days. Every 3 days the cycle is changing. That's why you can see that rodent or mammalian can have a baby more than usual. That's why we tried that when we use the estrogen, we found that it's quite interesting. The size of the stroke is become smaller than some of them actually have no stroke at all. Yeah, this is done in our lab, and then this is funded by NIH. The other also study that we do is we try to use the small molecule. In this case, we use the SOX3, these anti-inflammatory drugs. Then we put in the virus, or in this case, we use the adenovirus. So why we use this? we know that we can control the identifiers, and then they can infiltrate it into the area in the stroke, and the virus can leave this. A lot of neurologists or neurosurgeons asking whether we can put, for example, a stem cell in the ischemic area. What do you guys think? It doesn't make any sense. Why? Because it's ischemic area, there's very hypoperfusion, there's no oxygenation, and also the hyponutrient. So if we put the stem cell in that region, the stem cell will not survive. That's why until now there is no way that we will put, except we try to use what we call the stem cell induced trophic factors. So we collaborate with the UCLA, Clive Spencer, where we have the iPSC that we also contain the virus that can induce trophic factors. I don't showing in this slide, but we found that if we can use that, the trophic factor, the glial cell that we putting can release the growth factor, and then the is uptake by other cell nearby, and also the stem cell itself. Then they can growing, and it can survive more better than if we only putting the stem cell alone. The other case that also we do is we try to use RNA interference STAT3. Again, is an antioxidant, and as I mentioned to you, because we need to have the small molecule that can crossing the blood-brain barrier and also disperse to any area of the ischemic brain. And then we found that actually, we found that the RNA actually have very effective treatment for that stroke model that we have. And now also I just talking with doctors Prof. Rika that another candidate actually is in exosome itself, where we can also induce, because it's small molecule, where it's only below 300 nanometers. Then you can just injecting into the vein, and they will retrogradely coming to the ischemic area and then do the works. So I think in futures there is a lot of opportunity to use another small molecule, and then also again the small molecule is become best candidate for the neurodegenerative diseases in futures. I think I will stop my talk here, then I will entertain some couple questions that you guys have. Thank you.
Okay, thank you very much, Professor Irawan Satriotomo, for the talk. Okay, so let me briefly conclude what Professor Irawan already talked about. So I think from his talk we know that neural and also vascular communication is very important for the nerve system function. This communication might happen, for example, like through the growth factor. Growth factor secreted either by the neuronal cell or the vascular cell. And the disturbance of this communication may cause the neurodegenerative disease. And actually understanding this important communication between the neuronal and also vascular is very important. And it can be advantageous for us to design the therapeutic for the neurodegenerative disease, or also to improve the plasticity itself of the central nerve system. So, okay, so let us enter the Q&A session. So, Is there any question from the participant or the attendants here? Not yet? Oh, okay.
Thank you, Prof. Irawan, for the nice talk today. I will ask about estrogen. About the estrogen, we know from the lecture, estrogen it has a neuroprotective effect for our body, especially for human before menopause. Nah, in Indonesia, we know there are so many contraceptive like combination oral contraceptive, and we know we cannot use this COC, combinative oral contraceptive because we know that it can induce hypertension in women. So I think what's the explanation about this contradictive statement? The first one is estrogen gives us a neuroprotective effect, but the other side, birth control containing estrogen can induce blood pressure. That's from me. Thank you.
Okay, so
It's a really very good questions, yeah. So, again, in our body, the balance or hemostasis is very important. So, as I mentioned to you, that there is a very few incidents if we the women in the premenopause state, but again, probably the best way to do is actually with a very rigorous study to see how the estrogens play the role and how much that is can be maintained. If we and that's what the hormonal therapy begin. So if the hormone become, because you guys can, I'm sorry to say, women when we try flossing and then they know that they already have some problem with hormonal imbalance. Then if it's happening that, so the doctor can prescribe. But how it's correlated with the stroke and everything is very, we still doesn't know much the detail, so that we need to study more further. But now there's a couple study also in US that using that estrogen to treat the patient with stroke, and it's a male, because the estradiol can convert it into the I forget the pathways, but then can lead to induce the testosterone, and again the testosterone itself also can have the neuroprotective effect also. So this is the thing that we need to thinking. I think a larger study, rigorous study, and it is needed. And then a multiple center also need to be involved to see that how that this estrogen or the hormonal therapy can play the role, because there's a lot of study done in US or in Europe. They're showing there's no protective effect. Yeah, but actually, if based on our preclinical study and other preclinical study, there's a lot of proof. That actually we can do that. There is the discrepancy, because probably that you said before, that we need to have the standard, how much the estrogen to be needed and everything. The study that we did actually is double-blinded, and then this is done in multiple centers. University of Florida, University of California, and also the UNC, University of North Carolina. And then we found that this everything actually is showing the similar thing. Yeah. I hope it's answered your questions. Thank you.
Okay, thank you, Professor Irawan. Any other question?
Oh.
Terima kasih, Prof. Saya ingin bertanya, tadi ada model iskemik ya. Apakah dengan model iskemik dapat memperbaiki memori atau depresi berkaitan dengan teori neuroplastisitasku itu? Apakah bisa kembali 100% atau berapa lama waktu yang digunakan, atau mungkin dengan penambahan seperti apa, menggunakan antidepresan atau apa seperti itu?
This is also a good question. Interestingly, the stroke do not induce the cognitive decline, except if it's involving the hippocampus area. If you guys see this, interestingly, by the way, I also sending the information about the workshop that I did in Malang. You guys can learn the technique later on. What I do, I induce, I put fibers into the medial cerebral artery and included this area, and then after 60 minutes I take out that. So what's happening is there is no perfusion in this area, but when they do the hypoperfusion, when I took again after 90 minutes, there's still the hypoperfusion is still happening, and it's still maintained until the end. So the largest area in that model can up to, you guys can see, 50%. But in human, if you got stroke, like 50% of the area of the brain, you will be dead. But this is showing us that the rodent can be a very, what I call the plasticity of their brain is much more better than human. So if we do the study with the cognitive, probably with the maze and others, we don't see any changing of the cognitive. And you also found that people with the stroke doesn't have any problem with the memory. Mostly they have a problem with the locomotors. So how if it's involving into the hippocampus area, and this is larger, or they can get like in hemorrhagic stroke involving in the brainstem because it's very close with the blood vessel area, basal artery there. That will be can involving in the very specific area as well. That can happening with the cognitive decline or cognitive impairment will be happening in this area. So if we try to if it's happening in cognitive, can they be compensated? And the answer is yes. Again, but in human study, we never have the data yet for that purpose, because the area of the hippocampus is pretty close with the area of the brainstem and others. Then mostly the patient, if they have the stroke in that area, they will be much more dangerous rather than have in the cortical area. I hope this answers your questions. So the stroke do not involve the cognitive area unless it involves the hippocampus area. Yeah. Other questions?
Okay, other question? No, yeah. So maybe I have a question, Professor Irawan. So.
Oh, sorry.
Okay. Okay, so there's a question from online participant. Yeah. What are the marker for plasticity in the brain? Okay. The cell line for in vitro model stroke with OGD or oxygen glucose deprivation. So
Yeah.
what your opinion using this in vitro model, Professor?
All right. This is very, very good question too. A lot of study that I did with the many models. So the marker that rising during the post-injury mostly is growth factors like BDNF, VEGF. So you guys know why? Because it's the brain will try to recover try to develop the new neuronal or the precursor cell that needed this growth factor. So that's why all that growth factor actually is upregulated. Unfortunately, I don't seeing it. So in adult, we don't have it yet because why? We are done with the development process. But interestingly, I have a model what we call that intermittent hypoxia models. So what we do, we induce the hypoxia. Hypoxia is different with anoxia. It's totally different. What we do, we reduce the oxygenation to be half. Yep. So usually we breathe with 21% of oxygen. So then we reduce the oxygenations into 10%. So what's happening with that? If we give the oxygenation, the intermittent, so we induce the all the growth factor that we found. That is already published in comparative neurology papers. And then it's picked up by one of the postdoctoral fellow. He came to my presentation and they're asking whether they can use for human study. In this case, actually, the study done on the paralysis patient. You know, guys, that United States like to sending a lot of armies to abroad, and then when they come back, they have injury and paralysis and everything. Then these veterans really can do anything. So what we do, we giving 10% of oxygenation only just 60 minutes. Then, not 60 minutes, sorry, 30 minutes. And after another 30 minutes, we try to see the response from gastrocnemius as well as tibialis anterior. And you know what? That the EMG, also the activity become more higher than normal. And they feel something. And this Randy Trambauer now moved to the Harvard University, and they have the lab called Inspire. You guys can try to find it. So they're giving now the intermittent hypoxia to the patient like that, and then they also do with the bearing with the walking bearing treatment. So now the patient mostly cannot do anything now. It's starting to walk. So what the magic behind that? So we have proven that when we try to use intermittent hypoxia, we induce all that growth factor like BDNF, VEGF, even the signaling pathway like AKT, PI3 kinase. You guys name it. Yeah. And then it's also showing that receptor like TrkB and everything also upregulated as well. This is meaning that the model actually a really very good model to improve the. Then the next question that is about in vitro with using the OGD. Again, this is actually also a very standard model for the plasticity. And then I know probably Prof. Rika also knowing the Dr. Indra, that people that doing stem cell, including the Stem Cell and Cancer Institute. So if we'd like to make our stem cell become more powerful, more powerful meaning what? More powerful meaning that the stem cell can induce the trophic factor, the stem cell can induce a lot of microRNA, and that only the easy way is try to put the hypoxia. Is that correct? Yes. So that the answer. Why what we do in human study in vivo and as well as in vitro proven that actually dengan hypoxia can induce a lot of things. Yeah. So that again is depend on how we try to manipulate it, that timing point and everything. So yes, in vitro or in vivo and also the other study actually is still very good models that can induce the growth factor that probably can be applied, but for the human that got neurodegenerative diseases. All right, I think that there's other question.
Other
That
question? Oh.
oh yes.
There is no. Yeah, that's not a question actually, but I think we thank you very much, Professor Irawan Satriotomo. We are very proud of you because you are our alumni that have been as a leader scientist in the world. And actually, I have an appeal. Why don't we think about, because our government right now have been come into what they call it precision medicine era. Our Minister of Health have been launched what they call it Biomedical Genome Science Initiative last August, in the 14th of August. And there are some hospitals that have been 6 hospitals right now that have been appointed as the center to be a precision medicine. And one of it is about, of course, PON, the center, Rumah Sakit PON, Pusat Otak Nasional in Jakarta, is appointed as the Center for Neurological Disorders. But as we, as an institutional, have to deepen our knowledge, have to deepen our technology, that we also think about how we can contribute to the development of precision medicine, especially in the neurological point of view. Because as we know that, of course, cardiovascular disease is one of the killer disease among others, in Indonesia right now. So I'm thinking about why don't we have kind of research collaboration start from the group of neuroscience and looking for the certain neurological diseases, but the approach is omics, from the omics point of view. So we start from the genomic, proteomic, transcriptomic, proteomic, and metabolomic, because you have mentioned a lot about proteomic, metabolomic, as well as genomic. But the one point that we also have to do is transcriptomic, because this is very important as a functional in between. And I'm specifically come because your title is about crosstalk and about repair. So that's the point that I think we should do that. So Dr. Eureka is the PhD from neuroscience. I think why don't we think about crosstalk and repair in neurological disorders, and we can make a collaboration with Professor Irawan. And the approach is maybe again through transcriptomic, genomic, proteomic, and metabolomic. And we can start, of course, one thing that we just mentioned, exosome, from those plasticity regarding the content of the exosome must be different, because this is the real-time substance that can give at any time we take at the early, late of mid stage, it will be give you real-time picture of the situation. So this is, I think we should do that. And I'm thinking about why don't we can choose the best of the student and send to your lab to have the idea about research, neuroscience research. Yeah. So maybe we can have of course, the support from the government to do what we call it a mobility program. for the student as well as for the lecturers. So I think this is a very good start, and Professor Irawan is our adjunct professor here at the Gajah Mada University. So I think we not just stop in this room and hearing the very good presentation and research that Professor Irawan have done, but why don't we collaborate and think about the future and to support the precision medicine in neuroscience? I think this is a very big area that we can think and we can contribute to our country. Thank you very much.
Sorry, thank you.
Just one sentence. Because Prof. Roger Frutas is also our adjunct professor here at the faculty in infectious disease, and we have sent several students there in Montpellier. So I think we should learn from him that we cannot stop just giving lectures, but we have to continue. You are doing a lot of research on many kind of infectious disease, and we have to do that for neurological diseases. Maybe we can, because right now the government provides big grants, so we can do multi-center study, not only from UGM, but also with other universities. So I think it's good time and it's good point that we should do that, Professor Irawan, and also the student, and of course I appeal that also to Dr. Ulrike. Thank you so much. You want to comment?
Yeah, thank you, Sophia. No, I just have a short question, because there is, for instance, populational dimension in cancer. Different human populations don't have the same susceptibility to the same kind of cancer, and there is a variability. Is it true for the stroke? Is there a variability in susceptibility to stroke depending on the human population? And if yes, do you know why?
This is a big question too. So actually, we can see that the area or the stroke also involved in genetic background, and then we found also, for example, the lifestyle also changing a lot. So as I mentioned to you before, we only seeing the patient with the above 50, and now we saw that a lot of patients with the more younger ages also got a stroke, because probably they become obese, the lifestyle is changing, more stressor that they got. And we found also is a couple, study that done to see the different population, like for example in Japan. Probably the lifestyle there, as well as the food, that the consumption of the more better lifestyle is better than the other in Europe or in other countries in Asia. So the answer is yes. The background and also the demographic also changing the incidence of the stroke in that area as well. Yep.
Thank you.
Sorry, Professor.
Yep.
So what you are meaning is because the environment, lifestyle, and everything is so different. So might be this will affecting the regulatory point of view in the genome. So this is the epigenetic. Will play a lot of things in this pathogenesis as well as in the response of the treatment.
Agree.
Right?
Jadi genetic and epigenetic will changing a lot of things that can be interacting. Actually, I would like to comment as well what the Prof. Rika said, that I'm helping actually Dr. Setiaji from DTO, Digital Transformation Office, to do this genomic mapping, and I will meet Dr. Nijar and other people in Rumah Sakit Pon and discuss this. The problem is the cost of the NGS is 60 to 90 billion, and the region will cost you around probably 10 to 20 billion. But we don't have any resources for human resources, people that can do it, that thing. And then now we try to communicate with this thing. If the government will spending like 6, and it's true, 6 or 7 center, But if they are not ready with that thing, it is really very hard to do. The other interesting point is, Dr. Proprika just said about the precision medicine. Yeah. And in the US, now it's become more common. Before I came here, I got the annual health exam, and one of my doctors sent me the what they call Cologuard. It's big boxes, and then I just need to put my poop that, and then when they will sending to the company, and then we'll see that what the area that I mean, whether I have colon cancer or predictable for the colon cancers. And in the US now, it's become very easy to get the genetic exam. Like probably you ever heard 23andMe. If the company can give you the screening of like more than 30 genetic that you can imagine. From the 30, they will divide it to RA to become like very specific. And then you know where you're background from. Probably I'm part 20% from Europe, the other percent from Africa, from the other percent of Asia. So we can see the three liners. But not only that, and also giving you the information what you will be predict in the future. You guys know Angelina Jolie? Yeah. What she do? What she did? She did do the mastectomy before the cancer may be happening. So this is the powers of this science. This is the power of that omics area. Yeah, what the Proprika said about transcriptomic, genomic, metabolomic, lipidomic, and everything. Now everything actually is become one thing that probably I would like to mention is you guys need to master this skill set. Yeah, I have three staff. One of the staff that Dr. Yulis Hermanto now it's become he's done in Shira in stem cell with Isinya Yamanaka won the expert Nobel prizes. I also supervised Dr. Hendra Prabowo Suhendra. He did the master degree in the Karolinska Institute, and then one of my active staff is now become one of the expertise in the omics, Dr. Kristen Liesel in Thermo Fisher. And what I discussed with her is the same thing too, and I would like to share with you guys. It's the skill set that you need to have and knowledge that you need to have. So this is the thing that you guys need to consider for your own future. Exactly. I think that's
Prof. Irawan, just one thing.
Right.
You mentioned about the grant because we need a lot of big of grants to support, right? And I just come from Denpasar yesterday for Southeast Asia conference on genomic. Okay. Yeah, by the Minister of Health and several, and we got information. Actually, there are some opportunity. There are some certain foundation that provide us grants. Oh. So don't worry, maybe we can talk and then make just 2 pages of proposal, and they will provide the grants, and they can provide how many number of samples is times 13 million dollars for one sample. So if he needs to collect about 50,000 genomes. So if we can provide from stroke, say, or for neurodegenerative disease certain number, then we calculate times $30,000, make a good proposal, and then send it to him. I will give you the email address, and of course there should be a good start to have a collaboration with Professor Irawan for the research thesis for the students, and we can continue because it will be a never-ending story about analysis. Yeah, thank you very much. Di accent, Prof, karena jumlahnya nggak 50.000 sehingga itu bisa diproposkan.
Okay, this other question about during brain ischemia, what is the most effective in VU and microglial or neuron or other structures? And or it happens simultaneously, and how do hypoxia affect synaptic plasticity in the brain? Yeah, during the brain ischemia, actually, all that part of the NVU, neurovascular unit, also changing. The microglia, neuron, astrocyte, also other structure will changing it. They will try to adapt it, the process as well. And then it's happening simultaneously. If we get the chronic state, they will do, as I mentioned to you, they will become bigger, they become macrophages, they try to clean up the area. But after everything is done, they become resting glial cell and try to providing the trophic factors to the neuron, the surviving neuron there. This is interesting because I have a slide actually, but I don't bring it. In chronic diseases, in this model is called the ALS, amyotrophic lateral sclerosis, where that involving the phrenic motor neuron. Mostly the ALS patients die because of the respiratory failure that they have. And these models, the animal model, only have like 2 or 3 motor neuron left. The question is, how on earth that motor neuron can still maintain the breathing in that animal? Yeah, because of that, again, these glial cells, surrounding glial cells, try to providing the trophic factor to this surviving motor neuron. It's the same. I always make an analogy. If you have yours, your neighbors die, what you do? You bring a lot of things to them, don't they? It's the same thing too. So I always challenging with the neurologist or neurosurgeon whether is it good to keep the neuroinflammatory drug in chronic state. And the answer is not. If you remember, there is a larger study about using the minocycline for the ALS. What's happening? ALS patient, I mean, the patient actually is crashing more further. What we're thinking is that neuroinflammatory drug actually killing this glial cell that try to providing the trophic factor to this surviving motor neuron. So that how hypoxia affecting synaptic plasticity. Just, as I mentioned to you, the hypoxia actually will strengthen the synaptic plasticity. We do what we call APSC, but in this case we use the what LTF they call long-term potentiation. So during the hypoxia, actually the potentiation of the nerve fiber is much higher because it's much more profound. It's like you try to surfing this motor neuron. But after that, the interesting thing that is in steady state, the plasticity is still maintained, but the other trophic factor or others is maintaining this condition as well. So how about the vitamin D? This is very intriguing study. We always found that vitamin D become what I call that blaming vitamin in every single disease, including the COVID. You guys probably remember. They try to providing more vitamin D. Why? Because we found that the vitamin D in that all the patient or that kind of condition actually much more lower. But again, lowers concentration doesn't mean this is have a direct effect. So we need to be very careful when we try to make any decision. And the answer we still do not know. For example, people with multiple sclerosis, it's very few cases MS in tropical countries. But many studies found that in area that the sun doesn't have much there, yeah, like Icelandia or Norway in across of the globe, doesn't have much sun like that. And the case of the MS is more higher there. Then the people or the scientists try to connecting between that thing. But again, there is multiple process that we need to consider. But again, yes, vitamin D actually have some effect on that area. But how it's done, we still need to investigate it more further. I think that's what my answer. Thank you.
Okay, thank you, Professor Irawan. So, unfortunately, due to the time limitation, I think for this session of Professor Irawan, I think we can end it right here. So thank you, Professor Irawan, for the insightful talk. Yeah. Perhaps if you still have any question to Professor Irawan, we can have discussion maybe after this session. Yeah. Okay. So for the next session, for the second session of the talk, we have Professor Roger Frutos from the University of Montpellier. Professor Roger Frutos is the research director and also research director in CIRAD and also professor in University of Montpellier. He has his master and also doctoral degree from the University of Montpellier in pathology and parasitology. His main research, recently he published a lot of research on COVID and SARS-CoV-2, and then he has also collaboration with UGM in dengue research and also echinococcus research with the other university. So, Professor Roger Frutos, we invite you to the stage. Okay.
Thank you.
Okay.
Thank you very much.
Yeah, the stage is yours.
I take
Professor.
the mask off. It will be easier for you to understand what I say. I'm far enough anyway. It's useless anyway. So, oh yes, thank you very much. So the pointer, I believe, yes, and okay.
Sometimes there's a delay.
Yeah.
There's a delay.
Okay. Okay. Thank you very much. So, first of all, I would, I would like to thank all the organizers, the Faculty of Medicine of UGM, all our colleagues and friends to have invited me to give this presentation. And also I thank you to attend that presentation here and online. So thank you. And what I'm going to do is actually give some sort of overview of dengue. Okay. Globally, also the situation in Indonesia, and with respect to what we did also, and what we are doing, and what we are going to do, especially in collaboration with this faculty. So we have been working for quite a while on that topic with the Faculty of Medicine of UGM. So we have some current data coming from here, but also it's important to try to have a global view of the whole system. What is dengue? So you all know it's a virus, that everybody knows that. It's a virus, classical virus, so nothing to say much about it. The virus itself is very well known. The main point with dengue is that is the disease itself. Dengue is a complex disease at different levels. Well, first problem, there is no treatment. That's the first point. So basically, it's paracetamol and wait for it to pass. So no treatment. For the moment, no vaccine. either. Plenty of vaccines, the candidates are being tested, but for the moment, there is still no real completely efficient commercial vaccines. Is still under progress. Hopefully soon there will be one, maybe several, but for the moment it's not really the case. So already from the medical standpoint, it's quite a problem. And on top of that, we have 4 viruses. Okay, it's not one virus. It's 4 different diseases, 4 different viruses, and which are both dengue, but they act independently. If you get one, okay, you are not immunized against the other. In contrary, that's another problem. And we will go over that in a few minutes. There is also another point. In dengue, the rate of asymptomatic patient, like any infectious disease actually, is quite high. So depending on the virus, on the people, etc., it could range from 50 to 90% of asymptomatic. We don't even know they have dengue. Okay, they don't even know themselves they got dengue, but they still have the virus. And then we have the classic dengue. Then we are talking about the symptomatic people we know they have the disease. Okay, and so the classical dengue, the dengue fever, is 91–95% of the symptomatic people. So 91–95% of the 50 to 10% of people who show the symptoms, not of the whole population. And out of this regular dengue fever, a small number of people, 1 to 5%, okay, is showing a severe form of dengue. And part of them can die from it. So it represents a very small portion, okay? But it's very important. And dengue is rising, we're going to see that. But there is another major problem I just mentioned a few minutes ago, is that following a primo infection, okay, by any of the 4 serotypes, when you get a secondary infection, you have a higher risk of developing a severe form. Why? Because there is one phenomenon called ADE, so antibody-dependent enhancement, which means that once you have been infected the first time with one of the 4 serotype, there is an increased susceptibility because of the antibody produced against the first virus to the second infection. Okay, so the primo-infected are at a higher risk of severe form of hemorrhagic fever. But the point is, the primo-infected is not only the few percent here. The primo-infected is everybody. And we miss a lot of primo-infected because many of them are asymptomatic. So, and that's a point to consider, especially when we are going to discuss the situation of dengue in Indonesia. Okay, so as I said, I want to give an overview. So, like always, if we want to understand things, we need to take some altitude, I would say, to have a global view. And very often, it's important to go back to history and try to understand what happened. And what was the progress? So that's what we're going to do. Do a short history of dengue. What happened? Okay, what is the origin of dengue? Dengue originally, like many, not to say all infectious diseases, is not a human disease per se. Dengue, before infecting human, was a disease with a sylvatic cycle, so in the wild, on non-human primates, in other words, on monkeys. Okay, so mosquitoes and monkeys were maintaining the disease on monkeys, not initially on humans. And what is important is that this disease, this virus, is originating from here, Southeast Asia. Okay, in Indonesia, Malaysia, Thailand, it's the heart of the origin of the virus. And, but what happened? We just said that there are 4 different viruses. Actually, there is a 5th one, not on monkeys, so, but on human 4. And 4 viruses. Why do we have 4 viruses? Because over time, there have been 4 independent events of emergence from the sylvatic cycle to humans. Okay, at different time, perhaps most likely also maybe different place, okay, but certainly at different times. And like any population of living organisms, the viruses are organized in what we call a meta-population, so a population of populations. So within the meta-population of dengue, you have different populations which resemble a lot. They are very similar, but they are also a bit different. Okay, and depending on when and where the emergence occurred, a dengue virus that emerged in the human population, but it was not exactly the same. Okay, it was still a bit different, and that's why we end up with 4 different viruses. Okay, very recently you have exactly the same situation with SARS-CoV-2 and SARS-CoV, the first one, the one that gave the SARS disease in 2002 and 2004. It's one population of the SARS metapopulation of virus, and SARS-CoV-2 that is responsible for COVID-19, but is another one. So they are very close, but they are different. Here is the same story. And the point is that they moved from this sylvatic cycle to the peri-domestic environment, so in contact with humans, okay, especially in the river. So basically, that happened during the early human settlements, people going into the rivers in contact with those sylvatic cycle. So usually people in close contact with the forest. That is the origin. Okay, we don't know when. We just know it happened at 4 different times. That's it. But we have also to understand that dengue is an anthropogenic disease. In other words, a disease that it directly influences and related to the human society. And first point, and there's a big question mark, the first point you see is that the virus, I said it, is Asian, okay, it's from Southeast Asia. But the main vector is what? Is a mosquito called Aedes aegypti. Egypt. Even if you are not very good in geography, you know that Egypt is not really a Southeast Asian country. Okay? So we have the best vector of Southeast Asian virus, which is African. There's something wrong here. Okay? It doesn't really make sense. We have a secondary vector, which is Southeast Asian, is Albopictus. Okay? But what happens is that simply that these mosquitoes are actually anthropophilic. Means they feed, they take the blood meal, the females take the blood meal on humans, but they also live in the human vicinity, very close to the human settlements. In the human settlements. So that's very important. And this propagated simply because the mosquitoes, I mean, the virus is a bit different, but the mosquitoes propagated simply because of the trade. And it started in the basically 18th century with the big trade, you know, for spices, exotic woods, etc. But people transported goods, they transported mosquitoes as well. And today we do the same, simply the boats are a lot bigger and faster. But you have to understand that dengue initially was not called dengue. Dengue, when people didn't know what it was exactly, they called it the disease of the rivers and ports. That the people knew that they were getting the disease when they were going to the seaport and going into the rivers in contact with the sylvatic cycle, etc. They knew that because they brought the mosquitoes in contact with the virus. Okay, and you see, the first possible pandemic is very old. It's the end of 18th century, and with Batavia, which is Jakarta actually, Cairo, Philadelphia, Cadiz in Spain. So it's pandemic is not from today. It happened already in the past, and what the very, very key event in the dissemination of dengue is simply something that happened at the second half of the 19th century, is the opening of the Suez Canal. Because what happened in the Suez Canal, the boats were stopping in the Mediterranean Sea on the Egyptian coast, close to the entrance of the canal to get goods, people, water, food, and mosquitoes. They were getting everything at the same time, and by chance, it's just by chance, it happened that the population present in the Mediterranean coast of Egypt, which had no contact with Asia, when the canal was opened, was put in contact with Asia, okay? For the first time. And it happened just by chance that this mosquito population was extremely efficient in vectoring the dengue virus. It's not an evolution, it's not because they have never been in contact, it's just by chance, or bad luck, rather. And since then, dengue has just exploded. Okay? And so from now on, you understand something, is that it's just impossible to tackle dengue, okay, if we don't consider human activities. Because the dengue is developing because of our human activities, different kind of activities, but it is the human society that push the disease. It's very important to understand that, because the solution has to be sought within the human activities as well. Oh. Ouais. Oops, là. Went too far. Voilà. So, as I said, the role of human is very important because of in seaport you had high human density, the perfect condition with a lot of breeding sites for mosquitoes, a lot of movement. People went to the seaport, but it was just a stop. Then they had a contact with people living in the forest or inland, so it was easy to bring mosquitoes, to bring viruses, to bring infected people and make the contact with the mosquitoes brought by the big boats. And then the big boats were leaving and moving back the mosquitoes and the virus. So it's at that time it's also a lot of mobility, a lot of movements, which distributed the mosquitoes in different places. But it's not the end of the story. More recently, a lot more recently, what happened is the war in the Pacific during World War II was the key event for the spread of the disease of dengue in the Pacific region. And actually, the dengue was brought into Japan in 1942, with Japanese soldiers going back to Japan from the Pacific Island during the war. And during the war, you have a lot of mobility of people, very bad conditions of living, of course, a lot of breeding sites for the mosquitoes, and you have other things to do than looking for mosquitoes or virus, because it's a war. So it's a perfect condition for the disease to spread. And that's exactly what happened. And the four serotypes spread it all over the Asia-Pacific region. Okay, so you see, again, human activity is not the same. It's not business, it's war, but again, a human activity. South America is a very interesting story. It's completely different. The dynamic is totally different, but it's extremely interesting because the problem in South America initially was yellow fever. Okay, you have to consider also that yellow fever is not coming from South America. Yellow fever is originating from West Africa, just like Aedes aegypti is originating from Africa. So what happened is that the mosquitoes and the virus and some hosts were brought to the American continent during the slave trade. And the mosquitoes, virus, and hosts found a favorable environment, and the disease started in South America and developed in South America. It was a big problem. So after the war, the Second World War, I mean, they used a lot of DDT to kill the mosquitoes to try to solve the problem. That's exactly what happened. And you see 23 countries in Latin America, many countries in Latin America joined together to try to eradicate the mosquitoes with huge amount of DDT. Of course, they kill many other things, they polluted, etc. They somehow succeeded, or they thought they have succeeded, and so this happened in the 50s and 60s, ended in the 70s, and what happened? When people were doing trade with Asia, okay, but since they killed many mosquitoes, you see that in the 30s, in the 70s, most of the Aedes aegypti were eliminated, not completely, but mostly eliminated. They did some trade with Asia, so big boats brought goods from Asia to South America. They also brought mosquitoes. And the mosquitoes found the ecological niche empty, so it's a new home. They made it home, and they thrive. And what happened is that the mosquitoes developed, and with what they have, and what they had was dengue. So they killed the mosquitoes with yellow fever, and other mosquitoes came with dengue. So now in South America, you see, end of the 70s. Oh, I'm sorry. Let's go back. Well, end of the 70s, by DEV 1, in the beginning of the 80s, DEV 2 and DEV 4, and more recently, beginning of the 90s, DEV 3. So South America is also a hyper-endemic region with 4 serotypes circulating. And that's the important part of the lesson, and you never eradicate a population. So, the mosquitoes were still here, the yellow fever was still here, and now they have the 4 dengue virus plus yellow fever. So it's a very important lesson, is that eradicating is not the solution. You have to manage the risk and keep the mosquitoes there, because you know them, you don't know what could come to replace them. That's exactly what happened in South America. Now today, the disease is completely expanding. You see, this is the rise in the countries infected. So in the 2000s, in 2010, is going up, number of disease. So you have here, before the 60s, you have, or 70s, so you have the 4 serotypes in Asia. It's normal, Southeast Asia, that's where I come from. So it's normal. You have some in Africa, some in Latin America. Today, you have the 4 serotypes everywhere. Everywhere. So, Latin America, origin Southeast Asia, so it's already here. Latin America, 70s, 80s. Africa, is doing it. And Europe, it's started already. Europe is also a very interesting element, because dengue, that's what we call the tropical disease. Well, it's no longer tropical. It's a disease, but no longer tropical. We can eliminate tropical because of the mosquitoes that spread. For instance, the albopictus that spread in Europe, especially in France and Italy. Now we do have dengue, chikungunya also, and in Europe. And it comes from, the mosquitoes came from Southeast Asia, same thing with trade, international trade. Okay, now the mosquito is there, is in our place, he's there forever now, okay? And for instance, if I take the situation in France, in my country, in September 2010, we have the first 2 cases of autochthonous dengue. Okay, people who didn't travel. So it means that people, the dynamic is people get the disease somewhere, They come to France, they are bitten by the mosquitoes who was established, and the imported cases become then autochthonous cases, because these mosquitoes are going to bite people who live there and didn't travel, but they get the dengue. Okay, that was in 2010. This year, 2022, you see, it's very interesting. We had 165 imported cases only. Okay, if you, in 2019 we had 657, and in 2020 834 imported. It dropped considerably in 21 and 22 because of COVID-19. Because of travel restriction, people were not allowed to travel, so there was no importation of of virus. But even though, so that's why it's important, because even though in 21 and 22 there was a restriction in traveling, we had this year, as per October this year, 65 autochthonous cases. And if you go back, remember, to before COVID, to 19 or 20, you see 657 imported and only 9 autochthonous. 834 and only 14 autochthonous. This year, only 165 and 65 autochthonous, which means the disease, the virus, is spreading now locally. It's becoming some sort of endemic country. It does not rely necessarily as much now on imported cases. So it's really the disease that is spreading also. We have the same story with chikungunya. Talking about dengue, but chikungunya is the same. And so we have to consider something, you know, in the way to tackle dengue, is that with this Aedes borne diseases, there is a change of paradigm. We consider that a vector-borne disease is a disease that is transmitted by a mosquito from a human to another one. It's still true. It's still true. But at the global scale, this has changed completely. Because what happened? Because of the global economy over the last 30, 40 years, we moved the goods all over the world with big boats, but we moved the mosquitoes all over the world. The mosquitoes are now installed all over the world, north and south, depending on their capacity to stand the cold. Okay? And these mosquitoes are not necessarily infected, but they are vectors. And they are here. And what happened? Another trait of our modern society, well, except for the COVID-19 episode, it's a high mobility of people. We take the plane and we come from US, from France to Indonesia, etc. in 20 hours or so. And if you are infected, 20 hours, 30 hours, you are still viremic. You can still transmit the disease. And that's exactly what happened. The paradigm has changed completely, because now it's the humans who have become the vectors. We carry the virus to different places, and local mosquitoes just take it and infect local human populations. So again, we come back to the human society as a main driver. Okay? Whoops. So let's talk about the dynamic of mosquitoes and viruses. What we found out, for instance, in Vietnam, we had a study in Vietnam where we analyzed the different population of mosquitoes and the presence of dengue in the human population of the hospital and in the mosquitoes. And what we found out is that in different regions of Vietnam, we found mosquitoes, of course, and when we sequenced these mosquitoes and we looked at the haplotypes, the sequence, we found out that these mosquitoes were the exact same sequence we could find it in different countries around the world. The same. That was true for Aegypti, was true for Albopictus, exactly the same thing, and even for the virus, because we found out that the dengue virus on the 4 serotypes, okay, in the continental Southeast Asia, you have basically 2 different kind of populations. Some which were, I would say, endemic. They are present in one country, they stay there, they don't move, okay, and other viruses which were transborder. They would move to different countries. And it's always the same lineage, the same group of virus that moves from different countries. And this is for the four serotypes, sorry. So what happened is, well, when you look at the lineages, the phylogeny, if you want, of the viruses that stay in one country is different from the viruses that move. And the viruses that move from different places is different countries, so very, very long distances, is always the same lineage or the filiation, the daughter lineage, etc. So the mosquitoes don't do a long distance. Aedes mosquitoes, they do 200-300 meters maximum. A virus do 0 distance. It has to be transported. So how come they can go over 1,000, 2,000, 3,000 kilometers like that? Simply because they were associated to transportation of goods by humans. So some population of mosquitoes are associated to this movement, transportation of goods. And so you always find the same, of course, and they bring the virus they have. So you also find always the same virus that is moving everywhere. Okay, and we extended that work is currently underway, collaboration with UGM, so with our colleague or friend, my twin brother, Trimbaskoro Tungul Satoto, my former student who is now full scientist at Salatiga, in Salatiga, and a master student from UGM, Stephanie. We have tried to, plus my colleague from France, we tried to have a look at the phylogeny of the worldwide phylogeny. The worldwide, based on the data present in the database. Okay? We analyzed that. We retained only the full-length sequence. Because if you have the partial sequence are not always the same, they don't necessarily overlap, it's complicated. So we didn't take everything, we took only the full length, so that we were sure we can overlap and compare. Okay, we did the genotyping, the phylogeography, and for instance, what did we find? With Aedes aegypti, initially we found, you see, if you look a little bit of, if you know a little bit of phylogeny, when you see that, well, you see there are two populations, and this is the same, and this is the same, okay? Well, actually, these sequences, which would make a very large part of all the sequences present in the database, they were from China. They were all the same, exactly the same, 100% the same. So you could have only one, but we have plenty that was saturated the database. And actually, it was not Aegypti. We don't know what it was, but it's not Aedes aegypti. So we cleaned it, we removed it, and then we analyzed the remaining part here. And the point is, oops, I'm sorry. And that's what we get. We got a little bit of variation here, very little, and the rest is just a small break, but extremely small. And I would say two kind of clusters. I don't even say two clusters, two kind of clusters, because it's the same thing. The variation is very, very limited. This is on, sorry, I didn't mention that. This is on COX-1, so CO1, which is mitochondrial DNA. So we are looking at the maternal lineage. And what we find out, and you see these here, increased here, these are the other mosquitoes for comparison, other species. So basically, the conclusion for the we're doing that work at the moment. The provisional conclusion is what? Well, there is a lot of misidentification. For instance, half of the sequences were wrong. It's not Aegypti. But problem with the region sequence. It's not always the same, so it's so difficult. That distort the whole analysis. But what we see, we have only two very closely related population circulating worldwide. Actually, from maternal lineages, it's a maternal lineage. So basically, all the mosquitoes that circulate Aegypti worldwide is the same, it has the same origin. is one origin, is one population. After that, you have variation in the nuclear genome. That is different. This is the maternal. This is mitochondrial. So we look at the nuclear also, but it's not conclusive. It's not possible to conclude. First, because there is not too many sequences, so the information is less important, so more difficult to conclude. Most of the time you find the ITS2, and it's not good for genotyping because it's very highly conserved. It's not good for genotyping. And a lot less sequences are available. On top of that, CO1 and ITS2 sequences you find in the database, very often they don't even come from the same mosquitoes. So you cannot compare. It's a big problem. So the conclusion of that is that sampling and sequencing must be standardized to be able to compare and to do a very in-depth analysis. That's exactly why another collaboration with UGM, we are developing a specific map, a GPS-driven database to standardize everything, to formalize and standardize all the sampling and all the description of the virus, to be able to relate one single sequence in a virus with everything. The mosquito, the house, the container, the location, the people living there, etc., etc. That's what we are building now, to be able to have a good set of data, to be able, after that, to compare and have a good view of the dynamic. Right now, we cannot do it. To be the truth, it's a big mess. So, but we did it, but nobody else did it. We are the first one of doing it. So it again is a collaboration with UGM. And what the situation in Indonesia, because we did that also in Indonesia when Triwibowo did his PhD, but we found basically the same. We had a huge sampling over 43 locations all over, I'm sorry. Over Indonesia, from Aceh to Papua, so all over Indonesia. And we collected the mosquitoes and the virus, etc. What we found out is very simple. It was under the Ministry of Health with the Vectora project. But what we found out is the same population everywhere in Indonesia. You have one population, period. But interesting thing is that it's different from the population that has been described in 2013. But in 2013, there are very few samples. So is it because it's a mistake, it's not a GTI? Is it that the population was changed? We don't know. And we cannot compare because before this study, again in collaboration with UGM, there was no large-scale study like that, so we don't have any possibility of comparing the things. But it's only one population, basically. And we found basically the same thing with Albopictus. Very homogeneous. A bit less, but very homogeneous. Again, is the same population that circulate all the time because they're associated with the international trade, simply. Those which are not associated with international trade, but they stay in their place. So there is a huge bias and a huge influence of the international trade and the human activities on the kind of mosquitoes that circulate and the kind of virus that circulate. Okay, so now let's look at the dengue in Indonesia. But Indonesia is a hyper-endemic country for serotype. Okay, and the problem with Indonesia is that only when you look at the official data, only the severe cases, the DHF, are recorded. The regular dengue is not considered. But the DHF is just a very small part of the infected people. So basically, we are looking at 1 to 5% of the symptomatic patients only. So the symptomatic, which are not severe, are not considered. So the asymptomatic, which is the main part, are not considered either. So many people, they are completely ignored. So the burden is actually highly underestimated. There is a lot more dengue than what we think, but these people, light symptomatic, I would say, and asymptomatic, they don't put a burden on the hospital, but but they participate actively to the transmission of the disease. And that is very important. And if we ignore these people asymptomatic, we have to remember that the people who have already a primal infection, they are more susceptible to DHF. So we should not ignore them because they are potentially severe cases to come. And one solution we propose would be to isolate the infected people, consider them, okay, even if there is no big symptom, isolate them. Isolate them doesn't mean to put them in jail. Huh? It means to isolate them with nets so that there is no mosquito bite. And actually the viremic phase is considered to last 7 days after onset. If you isolate people for 2 weeks, which is not too much, you have a safety margin, and you're sure that when these people are again exposed to mosquitoes, they cannot transmit the virus, because when you put viremic people back into the population, you put a bomb in the population. You put a huge amount of viruses available for mosquitoes. And that we have to avoid, definitely. So what there is a need, the need is the detection, monitoring of DF, and the asymptomatic case. That's why we are, again, collaboration with GM, trying to develop at the commercial level some specific biosensors, cheap and very fast biosensors, to be able to do a large-scale screening and surveillance. Up. Voilà. So, still in Indonesia, and I'm finishing with Indonesia, we did a study, again with Tribaskoro, and in different islands of Indonesia, different cities, different islands of Indonesia, and we collected mosquitoes, and we also took the address of the people in each place who have been infected with dengue, declared dengue at the hospital over the last 12 months. And we mapped their houses. And what we found out is that it's not evenly distributed, except Bali. Denpasar, you have it everywhere. But with the exception of Denpasar, they are clusters. The virus, dengue is not everywhere. There are specific neighborhoods. And so not all neighborhoods are equally infected. For instance, we found out that unlike what people think, it's not the poor neighborhoods. The neighborhoods most affected are the upper middle class. Also, there are some cultural differences. For instance, in Lombok, you have a main difference between the Balinese population, so Hinduist, and the Muslim population, because the way of life is completely different. And the Balinese population is a lot more affected than the Muslim population. The main reason is that the way of life is different. The Balinese, they live in communities, so houses next to each other, one family per houses, but it's community. And they have plants growing, they have animals. So basically, they have water all the time. They offer a shelter for mosquitoes. When you go across the road to the Muslim population, Muslim neighborhoods, it's not like this. You have individual houses, very often no garden, so they don't rear the mosquitoes, basically. On top of that, in the Balinese, they are Hinduists, they all have shrines in the houses with offerings. But offerings is what? So a small container, a glass with water. So it's full of mosquitoes. And another place was more occupational, the wet markets. Because people, they have animals there, so they need to feed and give water to animals, so you find water, etc. Then you find mosquitoes. And one colleague who is here, visiting also, Eric Daudet from Rouen in France, is visiting at the same time, did some similar work in India, and he found exactly the same thing. He had exactly the same kind of results. Okay, so what we need to do is to identify the priority neighborhood to optimize the action and the resources. No need to waste all the money and the human resources, etc., in places where the probability to get a dengue outbreak is very low. Better to map, That's what we need geography. Better to map and put more effort where the probability is higher. That's what we need to do, and that's what we are trying to do. It's the project we develop now. Okay, but again, you see, with mosquitoes, very quickly, I don't want to take much time, we did that with Triboro and Tribascoro. We tested all the different methods of collecting, sampling mosquitoes. None of them are reliable. One method in one place will be fantastic. The same method in another place, zero. It's another method that is better. But the second method was completely bad, very bad in the first place. So you cannot rely on the method. It's extremely variable, so they are not reliable. So we have to forget about that. And so no correlation. And you know that WHO is giving some recommendation to use what we call the stegomia indices. So it's basically you count the mosquitoes in the house, in the container, you make an equation to have the number of mosquitoes, infected containers per 100 houses, etc. Give you an index. And depending on this index, the authorities will start mosquito control or not. Because this is supposed to be proportionate and to correlate with the risk of dengue outbreak. It's supposed to be an indicator. Actually, it doesn't work at all. We did that all over Indonesia again. We sequenced, we collected almost 70,000 mosquitoes to do that. The result is zero. What we found, it's not clear here, but let's see. I will pass. I went back, sorry. Voilà. Basically, what we found is absolutely no correlation between any kind of index and the risk of dengue. We calculated that with the incidence in each place where we collected the mosquito. Absolutely zero correlation. The only good correlation we found was, it makes sense, with the density of the human population. That's it. So we can use that. We cannot use that. It's so. What is left is vector control. It's the only thing left. No vaccine, no treatment, nothing. No predictor, no index. So only thing left is vector control. What can we do with vector control? Vector control. Voilà. So, sorry. Voilà. Chemicals. By chemicals is banned. Many places. On top of that, mosquitoes are resistant, so it doesn't work. So you can eliminate it. It doesn't work, and anyway, it's polluting, it's dangerous, and they don't work anyway, and they are forbidden. So, and you see, for instance, resistance in Indonesia. Everywhere. So forget it. It doesn't work. We have something else. Oh. I'm not good with that. But why it doesn't come back? Oh, it's something we call BTI, which is a bacterium. Actually, BTI stands for Bacillus thuringiensis israelensis, because it's a bacterium that was found in the desert in Israel. And it's extremely good for killing mosquitoes. In Europe, it's the only product authorized to do mosquito control. Only that. It's a biological control, so it's not polluting, it's environmentally friendly, etc., etc. It's very good. It works extremely well. There is no resistance. It's perfect. But there's always a but. But it works only on larvae. It doesn't work on adults. Okay, so you have to treat the breeding sites where the larvae is growing, and that's where we have the problem. Voilà. Because it's basically impossible to do it, because the breeding sites of these mosquitoes are a lot of small containers. You cannot treat all small pieces, hole in the trees, etc. That's where they breed. It's impossible. We cannot. So basically, chemical insecticides, they are forbidden, they are resistant. BTI works, but it's impossible to use it, because the breeding sites do not allow it. So we cannot use any kind of vector control on these mosquitoes, Aedes mosquitoes. So vector control is gone. What do we have? Some specific punctual vector control method, like the trap baits, like the Wolbachia, which has been developed here at the UGM also, repellent and nets, and the fogging to kill the adults if we can. But these are not large-scale system. It don't work on everything. Ovid trap, but you need to eliminate the natural breeding sites, otherwise the mosquitoes will breed, so it's very labor-intensive. Wolbachia, it works on Egypti, but not on Albopictus, because Albopictus already has Wolbachia, so the strategy cannot work. So you still have a good vector left. And efficient focking, yes, but he takes a very specific mapping of the, it's very difficult. And so basically, we have nothing left. And the only solution we have is what we call the integrated behavioral management. So the people themselves, to eliminate the population of mosquitoes as much as possible and prevent the population of mosquitoes from breeding and from developing. But it has to be done at the community level. Otherwise, it's too big, too large, it doesn't work. It works only at the community level. So I'm going to finish on that, if I can move the slides. Yeah. There is no single universal approach, it doesn't exist. We need to combine every environmentally friendly and sustainable approach we can have, and to eliminate not to eliminate the mosquitoes we saw in South America is not the solution. So what we need to do is to reduce the risk of infectious bite. That's what we have to do. So it's a matter of management. Okay, and we have to consider that the threat is global, but the answer is local. It has the community coming to Indonesia is at the level of the RT, actually. And in Indonesia, actually, it works very well. There is the Indonesian 3M procedure that has been implemented by the Ministry of Health. It works with one person to control per house. It works extremely well. So the good point of that strategy is that it works very well. The bad point of this strategy is that it works very well. Because since it works very well, people stop doing it. Once it works, they stop. Of course, mosquitoes come back. So the problem is sustainability. The only way to solve that problem is awareness. We need to what we have to focus on is awareness of the authorities and of the people. And that's what we have to do now. And it's a strategic approach using awareness to realize that dengue is an anthropogenic disease because of the human society. And the disease is caused by the virus, but the outbreaks are due to the society. And we need, for that we don't need, I'm sorry, it's the Faculty of Medicine, but I'm sorry, we don't need physicians, we don't need veterinarians, we don't need biologists. It's a societal problem. We need to discuss with sociologists, architects, urbanists, planners, modelers, policymakers, and more importantly, what I call the middlemen. Middlemen are people who have an influence on the population. So like, they could be leaders, wise men, teachers, but in my opinion and my experience, the person to work with are the religious authorities. Because whatever the religion, they have a kind of spiritual and moral power on people. People will listen to them. Whatever the religion, people will listen. And so they can bring the message and tell people what to do. It's easy to do, it's cheap, but it has to be done all the time. So we need to increase awareness. And in my opinion, we have to work with the clerics, with the religious authorities. And again, whatever the religion, they can forward the message at least once a week. So these are the person we should approach to implement that system. The solution is societal at the level of the community. Voilà. And so this is my final message. The solution is not technical. The solution is societal. The solution is awareness and the level of the community, the smallest community. RT is perfect level. And we need to talk with people, the middlemen, who can have an influence on people. And in my opinion, these people are the religious authorities. Again, whatever the region. So I thank you for your attention, and I stop here.
Okay, thank you, Professor Frutos, for your talk. So let me briefly conclude. So dengue, yeah. So it's at first it's a tropical disease, but right now it's already spread to many countries in the world. And this phenomenon is facilitated by human activities, high mobility, global movement. It's all affected this spreading of the dengue. And the solution and prevention of these dengue infection is not limited to one solution. So it's supposed to be an integrated from a lot of solution. One of it is the integrated behavioral management that need collaboration from all of the people, whether it's in the community, policymaker, and also the academia. So, okay, so I would like to invite from the participants to have a Q&A session with Professor Frutos. Is there any question?
Okay. Oh, yeah.
Okay, I have a question. The control of the mosquito is not chemical resistant. That the truth is fogging is not recommended again to use to prevent the mosquito, cause in the community still use the fogging to prevent it.
So
Yeah.
How can it work?
Yeah, that is mean it's not recommended again to use fogging to eliminate the mosquito in the community.
So how can it work at the level of the community? That's what you mean?
What?
Your question exactly at the community?
The chemical is resistant to the mosquito, but in the community we use fogging that use chemical to eliminate the
mosquito. Yeah.
Yes, that is still recommended or is not to use again.
But it's recommended, and on our experience, it is done. It is done at the community level, not by everybody, but it is done. The problem, and that's why we need education, we need awareness, is that that's what I say, it works. The problem that it works. The problem is not that it doesn't work, the problem is a problem of sustainability. Because as soon as people see it works, the mosquito, the number of mosquitoes goes down, they stop doing it. If they stop doing it, the mosquitoes come back. It's just like when people do a diet, ladies, people, as soon as you lose weight, you stop the diet. Then the weight come back. It's exactly, I'm joking, but it's exactly the same thing. The problem is that it works. So, and when it works, people say, okay, it's done. But no, it's a never-ending story. It's not something. It's not like I don't know, a fire or whatever. Okay, it's destroyed. You build it again. It's done. It's finished. It's never finished. So if you stop doing it, it will come back. So and that's why we need awareness, because it's a matter of education, and people have to integrate the fact that they have to do it all the time, even and especially if they don't see the mosquitoes. You don't see the mosquitoes, keep doing it. That means that you are doing it right. And as soon as you stop, whoop, it comes back. So, and that's where we need the awareness. That's where we need the middlemen to forward the message regularly. That's exactly, the solution is really there, is awareness, preparedness, and management at the local level, at the community level. If you do that at the community level, it costs basically nothing. It's extremely cheap. It costs nothing. It's a very few effort. It's just a matter of, I would say, attitude. People has to take the habit of doing things, controlling, etc. And that's it. Whereas the big action, you know, the big science, the big actions, at a very large scale, it costs a lot of money, it's very complicated, and it doesn't work. So, so yes, it works. It works. The problem is we have to convince people to keep doing it. That's the main problem. And Indonesia is the leader in that, huh? Because Indonesia has implemented the 3M. It works well. It works very well. It's a strategy to eliminate the breeding sites, etc. It works very well. So we should find a way to make it sustainable.
Thank you, Professor Futros. So I think she also mentioned about the insecticide. Yeah. So
you mean the chemical?
Yeah. So she mentioned about the chemical resistance. It's no longer
active.
Yeah. It's no longer used right now.
But it is still used. That's what the tricky point with the chemicals is, that they are banned for mosquito control, but they are still used because they are the same chemicals in agriculture. So even if you don't use the chemical on mosquitoes, they are exposed to the same kind of chemicals when people are treating in agriculture. And then they get resistant. And that's the problem. So we keep the resistance building. Even if we stop using that on mosquitoes, but on the rice field, on the orchard nearby, they spread. And then mosquitoes will be in contact, and the resistance will keep building. Again, it's a global issue, human activities. Health is not independent from agriculture. It's One Health. Actions done in agriculture have an influence on the public health. But we compartment, we consider it's different. So it has nothing to do. No, it's not true. They are related.
Okay, any other question?
Over there.
Oh.
Thank you very much for the chance. I'm still confused. How can the Aedes albopictus still infectious and spread the disease even though they have Wolbachia inside their bodies?
Simply because the population of Wolbachia don't prevent completely the virus, like what happens when we insert a Wolbachia in Egypti. So because the Wolbachia is not a vector control system, it's a virus control system. It doesn't kill the mosquito, in contrary, we need the mosquito to survive. And it prevents the virus from establishing. But the kind of Wolbachia naturally present in albopictus, but they don't seem to block the virus. Probably because it's a different Wolbachia, doesn't work the same, but that may explain also why albopictus is not as good a vector as Egypti. It's not as good vector as Egypti, but still is a vector. But nevertheless, the Wolbachia naturally present in albopictus do not prevent the virus from establishing and from being transmitted. Whereas it does when it comes to Egypti, in which we insert Wolbachia. And probably simply because it's different Wolbachia, I believe. It's not the same bacterium. So, and we cannot infect the albopictus with a new Wolbachia because there is already Wolbachia. It works on Egypti because there is no Wolbachia, but albopictus, no. So even if we control the virus in Egypti like this, but the virus will go through the next one, through albopictus, and still we will have dengue. That's the problem. But it works well. Wolbachia works well. The problem is that there are other vectors on which we cannot use it.
Thank you, Prof.
Okay, is there any other question from the audience? Okay, maybe a question from me, Professor. So what, okay, one of the solution maybe for this dengue infection is vaccine development. So what do you think about that? Because since right now...
Think of?
Vaccine development.
Mm-hmm.
Until now, there's no vaccine for...
Vaccine, I hope we will have one quickly. People are very cautious now with the vaccines because you don't know if you're familiar with the story of the vaccines, especially the one from Sanofi. No, what happened is that Sanofi developed a vaccine a few years ago, and they did some large-scale studies, especially in different countries, including the Philippines. And it happened that the Philippines, they also vaccinated children, and many children who have been vaccinated died of dengue. And so it was a big scandal, big problem, and what they found out is that the vaccines, when they vaccinated the people, the vaccines worked exactly like a primo infection. We have an ADE phenomenon I described. Therefore, the people vaccinated, instead of being protected, they were more susceptible. Hypersensitivity to the other, because it was only one serotype. Okay, and the people who have been vaccinated, they become more susceptible to other serotypes. And many children died. So they withdrew the vaccines, and everything stopped, and now people are very cautious in developing vaccines because they are very much afraid of the ADE. So it's slowing down the whole thing. So maybe the solution is to have a vaccine at the same time for the four, but it's quite difficult to develop. So it takes time, and we really hope it will work. But for the moment, it's still under assessment. Because there were a lot of hope before, people were rushing to put vaccines out, and after this problem with the Sanofi vaccine, everything went down, and people now are very cautious and really focus on the four serotype at the same time, and not only one, because they are very much afraid of seeing the same problem. And therefore it takes a long time, and I personally hope we will have one as quickly as possible.
Professor Frutos. Okay. Maybe one question. Okay.
Roger, long time ago, maybe several years ago, we are talking about global changes of global changes, climate change and global changes for this dengue virus. And you just mentioned today that you have evidence that the genotyping from Vietnam and also Indonesia, that you show us that it's not too much different.
It's the same, actually.
Exactly. So
it's not even too much different. It's the same.
So it means that in global movement, that we have to eradicate, right, the vaccine. So how do you think the best? Yeah.
So when I said, and I have to be cautious, it's the same maternal lineage. It means the same population, but it's not the same nuclear genome. So you have variabilities in the different population of mosquitoes, depending on vector competence, on resistance to something, to temperature, to whatever, because this is not encoded by the mitochondrial DNA, but by the nuclear DNA. So what we mean here is that simply it's the same population that circulates. So there's one origin, and it's always the same, probably because it's associated with large distance transportation. It's the same population that moves, but same maternal lineage. It's not the same population mosquitoes with respect to nuclear DNA. So the genetic background of mosquitoes, you will have different populations with different genetic backgrounds depending on the recombination, on the integration of different nuclear genomes. So some will be better vectors than others, that we know that. But they are still coming from exactly the same origin. We did that not to characterize the different population, that we have to target the nuclear genome. And that's what our project, we wanted to do the global sequencing, so full mosquito, plus the virus or parasite they have. But we placed that with 3 Boscoro and 3 Bowo to Gates, but they didn't accept project. It was not big enough for them. And so it's important. Our objectives was to see whether there were different populations circulating with different routes of circulation. Actually, no, there's only one. It's the same population and they circulate everywhere. But the same population in terms of origin, in terms of maternal lineage. There are different populations in terms of nuclear genome, nuclear DNA, and then in terms of Mendelian genetics, I would say. And these mosquitoes, even though they have the same maternal lineage, they have different phenotypes and different adaptation and different way of doing things. So that's why we developed the database. That's why we want to genotype the nuclear DNA in an organized way. Target real genes, important genes, and sequence always the same, and put that in the database to be able to characterize the different populations. But this time from the nuclear DNA. But for the moment, it's impossible to do that. The data don't exist.
Roger, there is a cross-kingdom regulation and cross-kingdom communication.
Mm-hmm.
Do you also see this kind of pictures of the cross-kingdom communication between mosquitoes and dengue virus and as well as human being?
But you mean is the word there is a specificity?
Yeah. No. What I mean is there are some publications mentioning that every cells, every living materials is secreting exosome, microRNA. That this microRNA, when come into the host, it will have something cross-kingdom, cross-communication.
Yeah.
Like what Prof. Irawan talking. So, do you think there will be something that can be taken from this?
Yes, it's possible. It's not a way that has been explored, but yes, it's definitely possible. Why not? Yeah, but we don't know what actually, and to speak the truth.
Maybe this is an era of something for the.
But it's something that should be explored.
Yeah.
For the moment, there is zero. But yes, it's somehow the idea with Wolbachia. It's not cross communication, but somehow because you infect a mosquito with a bacterium that will block the virus. Okay, so yes, it's somehow the same idea, but it's with a bacterium. Okay, could be maybe with some cross communication control, and why not? But one step for that is to make sure what is the diversity of the structure of the population. We know now that it's one maternal lineage, one origin, but we don't know yet what is the structure of these populations depending on the nuclear DNA. That we don't know, and we don't know either, so therefore what are the associated genotypes, what is the vector competence of different mosquitoes. We know there are differences, but it's not characterized.
So actually just a silly question. So you isolate the nuclear, and then you do the analysis of this nuclear DNA, and can you what? I don't know, because it will be mixed up between the cytoplasmic, mitochondrial, something with this? No?
No, it's a separate.
Separated.
Yeah, there are two DNAs.
Oh, okay.
You separate.
Oh,
You
okay.
separate.
Very easily. Okay.
Simply, the
Yeah, yeah.
mitochondrium is a bacterium.
Yeah, yeah.
It's a bacterial DNA, but this mitochondrium is what we call the cell heredity. Basically, is it transferred by the mother,
Yeah, yeah, yeah.
exclusively by the mother. That's what we call it maternal lineage. The male, the father, does not give this DNA. So we can identify whether we have 2 populations, 3 populations, how many origin. So we need it's the same maternal lineage, or there are 3, 4, 5 maternal lineages. Here, there is only one. It comes from the same place. Okay, but that's only the mitochondrial DNA. Then you have the nuclear DNA in the nucleus, and this nuclear DNA, then this one is susceptible to recombination during the reproduction, and this one followed the Mendelian.
Yeah, yeah, exactly.
And this one is a lot more complex. And it is this one which is associated with different phenotypes. So capacity to vector the virus, capacity to resist to the temperature or to something else, etc. Is on the nuclear DNA. And that, for the moment, we don't have this information.
Yeah. Thank you very much.
A pleasure.
Thank you.
Okay, thank you very much, Professor Frutos, for the talk. Okay, so I think we already at the end of this webinar. So to close today's event, we would like to thank once again to our distinguished guest speaker, Professor Irawan, and also Professor Roger Frutos. Thank you very much for the insightful talk. Hopefully this talk will start maybe the next collaboration with UGM. And also we'd like to thank to all of the participants who attend the webinar, either offline and online. And also lastly, as the organizer, we would like to apologize for all the shortcomings and the flow during this event. Finally, thank you for coming, and see you again in the next webinar. Good morning, everyone. Wassalamualaikum warahmatullahi wabarakatuh.
Do scuba diving on the coral reefs. And I'm coming from Jakarta. I was in Jakarta before.
Ayo, ayo teman-teman yuk.